oxygen comes into the picture. If you review the equation for aerobic respiration, the cells are not only using oxygen, they're also not only using glucose, they're also taking in molecular oxygen, O2, as part of the reaction. Now we're finally going to see this come into play. Oxygen atoms have a very high affinity for electrons. They are very electronegative, meaning they really want to suck in electrons. And so when the molecular oxygen comes into the cell, it goes into the mitochondria, and there's a binding site on this third cytochrome hydrogen ion pump for an atom of oxygen. The O2 has to break apart, so only one oxygen atom at a time plugs in there. But now that it's plugged in there, it's going to be able to suck in these two low energy electrons from the cytochrome ion, from the cytochrome complex. Now while it does that, it's also going to pull in a couple of hydrogen ions, and, well, you'll see what it forms here. That's enough to form a water molecule. The reaction that you just saw can be written like this: that an oxygen atom joins with two hydrogen ions from there inside in the matrix, in the inner compartment, plus two electrons, and that gives you a water molecule. If the whole thing happens twice, in other words, if we double everything, then we can write it like this: one molecule of oxygen plus molecular oxygen plus 4 hydrogen ions plus 4 electrons goes into 2 water molecules. And that way maybe is a little bit more accurate because it shows the oxygen in its more usual form, which is O2 oxygen molecules. And so that's the way I wrote it there. Now we saw something kind of like this in photosynthesis. What did we call it when we studied photosynthesis? Water splitting. But remember, it was run backwards in photosynthesis. When we studied photosynthesis, we weren't trying to get rid of electrons. We were trying to supply electrons to photosystem 2, because photosystem 2 was always giving out electrons, getting rid of electrons. And so in photosynthesis, it runs backwards. Water is split apart to provide electrons. Here it's going backwards. Water molecules are forming from oxygen as a way to get rid of electrons, as a way to suck up electrons. So we say that in respiration, oxygen is the final electron acceptor. It's the last thing in the electron transport system that sucks in the electrons. Whereas in photosynthesis, the water was the electron provider. All right, and so there goes that water molecule. So that was one NADH going through putting its electrons into the electron transport system. Let's look at an FADH2 putting its electrons into the electron transport system. The thing about FADH2 is it also carries a pair of high energy electrons, but its electrons are not quite as energetic as NADH's. For some reason, NADH's electrons are higher up on the energy ladder than FADH2 electrons are. And so as it turns out, FADH2 only has enough energy in its electrons to run 2 of the 3 cytochrome hydrogen ion pumps. It's going to run this pump and this pump, but not the first one. It just doesn't have enough energy to run all 3. And so what happens is, with FADH2, it delivers its electrons right to ubiquinone, so it bypasses the first pump. electrons. And so now it's going to run pump number 2. And then those get brought over to pump number 3. And so when those go through, hydrogen ions go through ATP synthase, we'll get 2 ATPs from the electrons in each FADH2. Now we're back to where we were a moment ago. These are now drained low energy electrons. That third hydrogen ion pump has to get rid of them. Well, again, it's going to be an oxygen atom. An oxygen atom plugs into the bottom of that cytochrome complex and grabs 2 hydrogen ions and those 2 electrons, and together that forms a new water molecule. If I click it here, it will just go through its cycle. Any questions on it so far? A neat thing. All right. I'll ask you a similar question that I asked you about photosynthesis. Part of this whole process is forming a hydrogen ion concentration gradient of high hydrogen ion concentration here and low hydrogen ion concentration there. And you need that gradient so this ATP synthase can make its ATPs. What things can you think of are contributing to that gradient? For instance, what things do you think of increase the hydrogen ion concentration here? The pumps, is that what you said? Yeah, each of these is pumping hydrogen ions up there. What is removing hydrogen ions from the matrix? Well, here's one thing, watch right now. The pumping, of course, but there's something else that's removing hydrogen ions. The water formation, yes. Watch here in a moment when we get a water formed that removes those 2 low energy electrons. Here we go. Here goes the oxygen. So to form that water molecule, it sucks in 2 hydrogen ions from the matrix. So that's also contributing to the gradient by lowering the hydrogen ion concentration there in the matrix. If you had a pH meter, where would you get a high pH, in the outer compartment or the inner compartment? Inner compartment, because high pH means low amount of hydrogen ions. So you get high pH there, and you should get a low pH there. And I'm not sure exactly what the pH numbers are inside mitochondria, but if I was taking a guess, it might be 3 or 4 pH here, and maybe 8 or 9 pH there.
So that third pump there, is that serving 2 purposes? I mean, it is serving 2 purposes, but it's not pumping in 2 directions, right? It's only pumping hydrogen out of the...
Correct. It only pumps hydrogen ions from the inner compartment, from the matrix to the outer compartment.
And when it creates the water, the oxygen, does it bond to that pump somehow?
Yes, yeah. There's definitely a binding site here for 1 atom of oxygen to bind right there. In fact, I think we'll see that happen in just a second. Well, so flipping back here to the overall equation for respiration again. This is where the water molecules are taken, just what you see up there on the computer screen. They come from, the oxygens in those water molecules come from those same oxygens that entered the beginning or entered the mitochondria. And again, it's something they weren't sure of. People wondered for a while if some of these oxygens in water actually came from the glucose, but they're actually derived from the oxygens that come from the process. And okay so far? All right, now, if you're in this class, you probably want to be scientists of one sort or another and not accountants, otherwise you'd be taking a business class right now. But nevertheless, I want to do a little bit of accounting, not in the tax sense, but in the ATP sense. So we know that each NADH can make 3 ATPs because it pumps 3 hydrogen ions, and every FADH2 can make 2 ATPs because they pump 2 hydrogen ions. I want to test to see if that actually works out. So we're supposed to get 36 ATPs. 4 of those 36 are made directly without involving any electron transport system, but 32 of those are made by the electron transport system from the high energy electrons there. All right, so let's total this all up. We got 2 NADHs from glycolysis. And we've got 2 NADHs from the transition step. This is per glucose that goes through the process. But most of them came from the Krebs cycle. That gave us 6 NADHs and 2 FADH2s. All right, and we know we're supposed to get 3 ATPs per NADH. And 3 times 6 here is 18. And we have 2 ATPs made for every FADH2. 4 ATPs. Okay, so we add these all up. We get 34 ATPs, which seems wonky because I told you earlier that we get 32 ATPs from the electron transport system. And so that's kind of the mystery here. It seems like we should be getting 34. Why do we only end up with 32? You know, it's like one of these accounting firms that cooks the books for these big corporations so they can cheat at their taxes. Somehow it's not quite adding up. Well, people were curious about this. You know, where's our missing 2 ATPs from the electron transport system? And had an interesting answer that in a way relates to some of the concepts from the energy metabolism chapter. And so let's take a peek at that. Here's this big picture of where those NADHs and FADH2s are generated that feed into the electron transport system. And notice that the NADHs and FADH2s that are generated in the mitochondria, those electron carriers don't have to cross any membranes to get to the electron transport system because they're already made inside the mitochondria. But the 2 NADHs that are made here in the glycolysis step, glycolysis takes place out in the cytoplasm. And so those 2 NADHs do have to cross some membranes to get the electron transport system. And that's where there's the rub, so to speak. These arrows represent the glycolysis part, step of aerobic respiration. And so those NADHs have to pass through membranes to get the electron transport system. Let's watch them do it here. Here they come from the glycolysis step. The problem is this: the Krebs cycle and the transition step have already generated a whole bunch of NADHs. And so the concentration gradient is such that there's a large concentration of NADHs inside the mitochondria, a lot higher than you find outside the mitochondria. And so by diffusion, which way do NADHs want to move, out or in? Out, right. And so it's a non-spontaneous reaction to move these two NADHs from glycolysis into the mitochondria. And non-spontaneous reactions always require some energy input. And so here's the way the mitochondria deals with this. Remember that FADH2s are also electron carriers, but they carry electrons at a lower energy level. And so what it does is it sends out two FADs to pick up those electrons from the NADHs. And so the electrons are actually going to lose some energy going from this one to this one, because this one's electrons are just intrinsically at a lower level. And so the energy that those electrons give up to go from here to here is enough to make the non-spontaneous reaction spontaneous. In other words, the electrons are passed to an FADH2 to get them inside. And the same with that one right there. It can't come inside, and that would be non-spontaneous, and so it just passes its electrons to an FADH2 like that. And so if we consider that now, these two NADHs, even though they're made initially as NADHs, get changed into two FADH2s, which you only get 2 ATPs for. And if you factor that in, then it comes out to the 32 that we're supposed to get.